Oh yeah.
Okay. Erase that greedy one board. Alright, so part 2 of chapter 15. So part 1 we looked at how information flows. You go from DNA to RNA to proteins. We're going to talk about the transcription process today, the translation process tomorrow. We looked at DNA, the genetic code, how it's a triplet. What's another word that we use to describe the genetic code? Starts with the letter C.
Codon.
A codon, okay? And a codon codes for one amino acid. How many different amino acids are there?
20.
20. So the code is a triplet, which means there's 64 combinations. How many combinations code for an amino acid?
3. Oh.
Out of 64. How many code for amino acids?
61.
61.
Oh, that's what you're saying.
Yeah, sorry. And then the 3 code for what?
Stop.
Stop. What's the codon that always starts every single protein?
AUG.
AUG. All right, so that was kind of...
So wait, that starts or stops?
Starts. AUG starts.
We have to know the 3 ones that stop.
No. You just need to know that there are 3. Okay, so today's objectives, we're going to look at RNA polymerase in detail, what it looks like, how it works. Again, you'll see the form follows function. We'll compare prokaryotic transcription to eukaryotic. It's a little bit different. We're going to talk about the process of transcription and how initiation involves some promoters as well as some terminators, and then break down the 3 steps: initiation, elongation, and termination. So RNA polymerase and its function. Here's kind of a cartoon diagram of it. If you were to cut it in half and take a look at it, you can see that there are 3 tunnels or 3 openings. Okay, they call this part the jaw. The DNA, the double helix, enters, and then inside is a wedge, if you will, that exposes or unzips the DNA, so we have a strand that's, that we can copy. It copies the template strand. The coding strand does not get paired up. It's just there. We have another opening where nucleotide pairings come in. So just like loose nucleotides floating around in the cytoplasm enter, and if they match up complementary, then they form a bond. And then we have an exit where both the DNA and kind of the mRNA, the new mRNA, exit, and you'll see that hopefully. But it's a very large and globular protein. We know that through X-ray crystallography. Where the channels intersect, that's basically kind of the active site. This is where phosphodiester bonds form and unzip, and the base pair complementary. Now the function of it, DNA fits into one of the enzyme's channels. Like I said, remember DNA is double helix, so you have to kind of unzip it to expose a single strand. That's the template strand. The strand that's not used is the coding strand. And so now the question is, how does the enzyme and the DNA come together? Well, they come together through a series of promoters. So let's just talk about prokaryotic transcription because it's different from eukaryotic. So with prokaryotic, RNA polymerase exists in 2 forms. You have a core polymerase called the RNA polymerase, and then you kind of have, when it bonds to something called sigma, then it becomes a hollow enzyme, which means whole enzyme, a functional enzyme that does what I've just described, unzip DNA, bind to it complementary. Sigma has to bind to RNA polymerase in order for initiation to begin. So when you just have RNA polymerase by itself, it's just the core polymerase. But then when sigma binds to RNA polymerase, now that enzyme is whole, it's functional, we can start the process of transcription.
What one is this on class?
What?
What one is this on?
This is part 2.
The one you just told me?
Yes. Yep. So this diagram cartoons it. This is the core RNA polymerase, and when sigma binds to it, now you have the hollow enzyme, and now it can interact with DNA. DNA cannot interact with the core enzyme without sigma. And you can kind of see sigma is the one that's interacting with DNA. So sigma by itself can't do it, core enzyme by itself can't do transcription, they have to be together in order to start transcription. So then in this cartoon diagram, here's my new mRNA. Here's the DNA strand. It's just rudimentary. But here's the sigma factor. And if it binds onto it, as you can see here, then you can start the process of transcription. Okay, so on the DNA strand, in order to start to read a gene, you have to have two things. A promoter, which promotes that the gene be read, and then a terminator, which means that we need that this is the end point. This is the end recipe for this gene or protein that we are making. So the promoter is the start site, and it's usually a series of nucleotides that is recognizable by the RNA polymerase with the core enzyme in this sigma subunit. Okay? The terminator will be at the end, and it's the thing that ends the process of transcription. So when the RNA polymerase reaches this, it recognizes that it's a stop codon, and everything just falls off, and now you have your mRNA. Everything in between the promoter and the terminator, we call that the transcription unit. That's the thing that's being transcribed to make mRNA, which will be translated into a protein. So there are certain sites on the DNA, like I said, that code for the promoter. In prokaryotes, it's usually called the 35 box and the 10 minus box, or the tata box. So like this region right here is the promoter that the core RNA polymerase recognizes, and then sigma will come in and latch to it, and it fits perfectly with the sigma, and then it starts to transcribe it. So I think, you know, on your exam, I might have a question that says, like, which one of these is a promoter? And I'll have the tata minus 10 box. Or, you know, that's the answer you want to choose. But I could, like, trick you and say the tata minus 35 box. That's incorrect. So it's the tata box. But when DNA is, you know, being transcribed, it goes in a certain direction. Okay. Bases that are upstream receive a negative number, and then bases downstream receive a positive number. So this would be upstream, and this would be downstream.
Wait, which one's upstream?
Past here. Because this is minus 10, this is minus 35.
Okay.
So promoter sequences are upstream of the start site. They're not transcribed. There are two 6-base sequences, the minus 35 box and the minus 10 box, the tata box, which is more recognizable than the minus 35 box. So sigma binds to this. This is a promoter in bacteria. So it recognizes 35 nucleotides down this sequence and 10 nucleotides down this sequence, and this is where it latches on. So sigma is very specific to these sequences. So what is the function of the promoter?
Start
Transcription.
You have to start transcription, and what binds to the promoter? Sigma. Very good. What's the function of the terminator?
Stops.
Stops the transcription. Okay, so sigma binds to that specific promoter, the minus 35 box and the minus 10 box. That's what it recognizes. What's this whole structure called when the sigma binds to the RNA polymerase? The polenzyme. Good. Whole enzyme. Very good. Sigma, not RNA polymerase, makes the initial contact with DNA, and you guys have already recognized that. So sigma is a regulatory protein, controls transcription, tells RNA polymerase when and where to start transcription. DNA opens up at the minus 10 box, and when it starts to open up, then you have monomers, which we call ribonucleotide triphosphates, or NTPs, which you saw on the previous diagram. They pair complementary to the template DNA. What's the difference in RNA between DNA for nucleotides? The U instead of a T.
Wait, to use an RNA?
U's are in RNA.
Okay.
Yep. So all unicorns can get all. A pairs with U, C pairs with G. Yeah. Okay. I was trying to think of the other saying that you guys had come up with. For DNA, it was like something about Tom Cruise. No, you guys don't even remember.
I don't think that was us.
Yes, it was. 'Cause like made a mental note about it. Okay, so after initiation, we have this process of elongation where we are pairing those NTPs complementary to the DNA strand. So RNA polymerase moves along DNA in a 3 to 5 direction so that RNA is made in 5 to 3. Everything's always made in 5 to 3. Okay. RNA polymerase leaves the promoter, sigma breaks away from the promoter. To give it a little bit more space, it gets unzipped, and now we just start base pairing complementary. Note the structure of RNA polymerase with its channels. Okay, so this is a diagram in a college bio book that I have, and I think it's just really, really clear. Here are the 3 openings, okay? Here's the sigma interacting with the DNA. You can see that this little hook latches on to the -10 ta-ta box, and then it's got another structure that interacts with the -35, so it's latched on. This is the promoter. All right, now we have a sequence that we need to read. So then DNA starts to move, and there's literally a little wedge inside the RNA polymerase that unzips it, exposing the template strand. Here's the coding strand. Our NTPs come in, they start to bind to it complementary, and there's another opening where the new RNA strand is made. DNA gets unzipped and rezipped as it passes through RNA polymerase, and then when it reaches that terminator, everything just kind of falls off. Sure. Good? All right. So question, in what direction is RNA synthesized? 5 to 3. Very good. Now, what's the final step in transcription? We've talked about initiation, elongation, and termination. So termination does occur at specific sites. We do have some stop sites. When the RNA polymerase, the whole enzyme, reaches that stop site, phosphodiester bonds no longer are formed. We have this DNA-RNA hybrid. What they do is they just fall away from each other, okay? And then DNA rezips back up. Bases that make up the stop code have this unusual property, and that's why they're considered stop codons. As soon as the site is synthesized, RNA sequence folds back on itself and forms a short double helix held by complementary pairing. Basically, it's going to form a hairpin structure like this. So that way when RNA polymerase reaches it, it can't go forward anymore. It hits a bump in the road. You can kind of think of it as a railroad track. And that's how it falls off. So the results of this hairpin structure disrupts that interaction between the RNA polymerase and the DNA, and as well as the RNA transcript. So all 3 things, and then literally a physical separation between everything. So to show you that, here's a diagram where we have our DNA strand. Okay, this is the strand that's being copied. So that's the template, and we have our coding strand, which is just there. Things bind to a complementary, as you can see it's moving, and constructed in a 5 to 3 direction. So now we reach a termination signal where it starts to form a hairpin loop structure, and RNA polymerase no longer can stay on it and falls off. It's usually made up of a bunch of A and U pairings, if you will, as you can kind of see here. But there's that hairpin structure, and then it falls off. Now, in prokaryotes are different from eukaryotes. How are they different? Eukaryotes have a nucleus. Okay, prokaryotes don't, and this is the reason why transcription is coupled with translation. There's no nucleus. So prokaryotes, transcription happens in the cytoplasm, translation happens in the cytoplasm. They basically happen simultaneously. I guess not at the same time, but transcription starts and then translation starts immediately after transcription is done. So prokaryotes don't have that nucleus. As soon as it's transcribed, it gets translated. mRNA in prokaryotes is organized so that genes are clustered together. So here's an example where we have genes clustered together that work very closely together. We have our promoter, Okay, and we're going to talk about operators in Chapter 16. But an operator turns on and off genes. Okay, so in this case, the gene is on. And then we have a series of genes that are literally together that help carry out similar functions. Structural gene for beta-galactosidase. That's an enzyme that helps break down galactose. But then we also have an enzyme that, I don't know what permease does, but I'm sure it's very similar to galactosidase. Galactoside transacetylase. So it's probably a assembly line of enzymes that take galactose and break it down even further. And so they are paired close together because translation will happen immediately after transcription. So in this diagram, we have the DNA, okay? We have multiple RNA polymerases making mRNA, but then you can kind of see ribosomes have already attached to it, and hopefully you remember that ribosomes make proteins, and they're making proteins from the mRNA. So they're zooming in on the ribosome. Hey, we're already forming our amino acid polypeptide chain to make a protein. Any questions on this slide? So they're coupled. As soon as one's done, the other one starts. Now in eukaryotes, it's not like that because transcription happens in the nucleus, and then that mRNA has to travel out of the nucleus to the cytoplasm to a ribosome. And so eukaryotes is obviously a little bit more complex. So eukaryotic transcription, very similar with a few exceptions. So to compare and contrast prokaryotes to eukaryotes. In prokaryotes, we have a single sigma protein that binds to the promoters and starts the transcription. In eukaryotes, it's not sigma. They're called basal transcription factors.
Wait, so earlier when we were talking about
That was all prokaryotes. Yep. Eukaryotes, it's not sigma, basal transcription factors, and I'll show you a diagram of it. Another is the RNA polymerase are a little bit different. They recognize different sites. So RNA polymerase II promoters are located at a -30 box. Eukaryotes also have transcription factors. So a transcription factor is, let's just say that this is my cell, and this is my nucleus with the nuclear pores, all right? And here's my double helix DNA. Let's say that a cell is like going through some development, okay? So it's got to turn on some genes because maybe it's going to move or migrate or form a new protein. Maybe it's going to form a new hormone because this individual is going through puberty, right? So it receives a signal from an outside hormone, okay? And all of a sudden it starts a chain reaction of molecules that interact with one molecule and interact with another, and then it enters the nucleus and says, okay, we got to find that gene to turn it on. So it literally attaches to the DNA and will turn on that gene to produce, more of a hormone for a cell. So this is a signal transduction pathway. Received the signal, activated some other molecules inside, which entered the nucleus to turn on that gene. So these would be considered transcription factors, things that bind to the DNA saying, hey, I'm turning this gene on, it needs to be read, we got to make this protein right now. It's really, really complex, okay? So you can kind of see here, it's my DNA, right? And we have a bunch of these basal transcription factors being bonded or interacting with the DNA, so that way the RNA polymerase can recognize, hey, this is a gene that needs to be turned on, I got to bind to this region and then start reading the DNA loop. So it's a bunch of proteins that bind to it, manipulate the shape of it a little bit to expose it to the RNA polymerase. So, bottom line, eukaryotes use basal transcription complexes. Prokaryotes use sigma. Sigma is way easier. Basal transcription, way more complex. Okay, some other differences. In prokaryotes, there's just one RNA polymerase. In eukaryotes, there are 3 different RNA polymerases. So, in eukaryotes, we have polymerase 1, 2, and 3. Pretty original in their names. 1 helps make rRNA. rRNA is responsible for making a ribosome. RNA polymerase 2 transcribes mRNA and small nuclear RNA. We haven't talked about small nuclear RNA yet, but we will. But it helps transcribe mRNA or help make mRNA. And And then we have RNA polymerase III, which is responsible for making tRNA, which stands for transfer RNA. tRNAs are responsible for carrying amino acids to ribosomes. And remember, a ribosome makes proteins. And how does it do that? Well, it links amino acids together. So, RNA polymerase makes rRNA. RNA polymerase II makes mRNA. RNA polymerase III makes tRNAs. So they all will eventually interact with each other. Okay, in prokaryotes, terminators are well defined. In eukaryotes, not well defined. And I think I'll talk a little bit more about that. In prokaryotes, mRNA is not modified. So when they make mRNA, they have their genes ready to go. Gene 1, gene 2, gene 3. They're all clustered. They all have they all work together for a certain job. In eukaryotes, looks like this. Here we have mRNA, and they're going to say exon 1, intron 1, exon 2, intron 2. So what I mean by exon is that certain parts will be spliced and deleted. It's kind of like making a movie. You know, you take a bunch of shots for your movie, but you don't put all those shots in your movie. You pick the best clips. So that's kind of what eukaryotes do. So we'll talk more about mRNA modifications. So let's take a closer look at that. mRNA modification. So, RNA is made, mRNA is made by polymerase II, but it is modified. And one modification is that on the 5' ends, they're going to put a methyl G cap. And what the methyl G cap does is it's a signal to say, hey, ribosome, bind to me here. I'm the start of the code of what needs to be read. Okay, so it kind of acts like a mini promoter for the ribosome. The end of the mRNA is basically a bunch of A's, adenine. So they call it a 3' poly A tail. And it's either between 1 to 200 A's for nucleotides, just latched onto the end. So if this is my mRNA, where they have all the genes and the exons and the introns, then at the very end, they're just going to pull out a bunch of A's. Now the reason for that is as soon as mRNA leaves the nucleus, enzymes start to break it down. And so the poly A... Hello, Aaron. Yep, no problem. Okay, bye. You two have to go to the office. You're not in trouble.
You look like an idiot when I walk. I better put coat on.
Okay.
You need a little belt.
So as soon as the mRNA leaves the nucleus, it starts to be broken down. So these poly A tails that don't code for anything act like a distractor or a lifespan expander. It allows the mRNA to live, if you will, long enough before it is completely broken down by an enzyme. So it prolongs the degradation by ribonucleases in the cytoplasm. But there's more modification to it, so we'll talk about that here. In eukaryotes, mRNA does get spliced. So here's an analogy. I have a bunch of gibberish with some words. Can you guys figure out the message? Right, yeah. And so what mRNA does, pre-mRNA does, is it removes all the gibberish. So that way your final product is like the prokaryotes where all the genes are lined up together. So the gibberish are the introns, because they're intervening with your sequence. And the exons, which you would think would be, excised out or cut out, that's not true. Exons are expressed. They stay. How confusing is that, right? So introns are out, exons are in.
That's good. Easy to remember.
Yeah. So here I have my RNA, my mRNA with my exons, my introns, introns. And what we're going to do is we remove the introns. And then, so we cut out, we splice, we re-glue back together exon 1, exon 2, exon 3. There's my poly A tail. There's my 5G cap. That is a signal for the ribosome to latch on. So the ribosome will latch on into the translation and then just start to read these genes. How are introns removed? They are removed by SNRPs, okay? Small nuclear ribonucleoprotein particles. These SNRPs help form something called a spliceosome. So here I have an exon, here's my intron, here's my exon. So these SNRPs latch on to the beginning of the intron and the end of the intron, and then they fold it. They cause the mRNA to fold. And when the spliceosomes meet in the middle, they cut and then re-glue the two exons together. Okay, so SNRPs cluster together, they form a larger complex called a spliceosome. So when they meet, spliceosome, they cut, they cleave, they re-glue. They say it forms a lariat. Lariat is like a lasso, if you will. But they re-glue the two exons together. There are no rules that govern the number of introns that can be found in a gene. Introns, they're there. We don't really exactly know why they're there, so it's just kind of one of those mysteries. But we do know that they get cut out by SNRPs. And here's a picture of a SNRP with another SNRP. They work together, they form a lariat. Then when they meet, a spliceosome, which means they cut it, and then they re-glue it together. And now this excised intron is floating away. It will be degraded by enzymes. And now we have our two exons together. We call this pre-mRNA. And then when it's modified, we call it mature RNA, which means it's ready to go out into the cytoplasm and be read by a ribosome. Yep. No. Okay, question. Which parts of the RNA are removed?
Introns.
The introns. And which enzyme removes them?
Yes.
What then are
The
the SNRPs?
SNRPs, and then the SNRPs form spliceosomes. Okay. All right, so at the end, I have some transcription questions. If you haven't handed in questions from last week, do so. I'm going to get to them today during my Earth Science Mineral quiz, because I don't think I got everybody's. But short answer, there's some multiple choice, more multiple choice, more multiple choice.
Oh my god, I don't like this. I don't like these.
But they all relate to what we just learned. Like I picked them.
I know, but I started to go back in my notes and look when I have some of my notes on my paper.
Oh, I see. I'm sorry. But yeah. Okay, the rest of the time is yours to work. If you have questions, don't hesitate.